I would like to invite to the stage to Pedro Serbastião, which is Technical Director of Asfertglobal. So welcome. The green one and the red one to back.
Okay. So good afternoon, everybody. So I'm going to present a study that we made in our research center about a bacillus strain that we have on our collection. So the biggest problem in agriculture nowadays is climate changes, and more than the increase of the average global temperature is these extreme temperatures periods that we have that affect crops, but also drought is a big problem. And in the future, most probably, drought will be an even higher or bigger problem. There are studies that say that in 2050, more than 50% of the arable land will be on a drought situation. And that will affect, of course, not only the crops, but even the soil microbiome. So to deal with these problems, one of the best tools probably to mitigate these problems with this abiotic stress can be the use of biostimulants or the microbial biostimulants. On those products, the multifunctional plant growth promoting bacteria can be an interesting solution. So these organisms can help the plants to overcome the stress in several ways, by producing phytohormones, producing enzymes like the ACC deaminase, producing metabolites that are adapted to improve the tolerance to the stress, exopolysaccharides, and once again, some antioxidant compounds. So like I told, the study is about a Bacillus valezensis strain that we have on our collection that we named BS36. He has the particularity that it was isolated not from the soil, but from a tomato leaf on the fields. And yeah, so on the study, we had two objectives. The first one was a study with omic analysis when we combine genomic, proteomic, and metabolomic data to see or to try to detect if some genes and compounds were inferred regarding the PGP activities. As a second objective was an assay on our trial station to see if this bacillus strain could enhance the maize tolerance against drought. So the multi-omic analysis are a good solution, a strong solution, to get more insights about the modes of actions of these type of bacteria. So on our genomic analysis, we made a complete genome sequencing using the NGS technology, and then we made some genome mining using some softwares like Rust or anti-smash. On the proteomics, we used the liquid chromatography, and on the metabolomics, we utilized the Fourier method. Well, the results we got was about 4,000 genes detected and around 2,000 proteins detected. Also, due to the accuracy and the high resolution of the Fourier method, we detected 730 metabolites. So, try to exemplify what we did. So like I told you, it's a very interesting tool to determine if we have traits or not on these type of bacteria regarding the biostimulant function or the stress alleviation. For instance, after the genomic and proteomic analysis, so we found these key traits on this strain, like phytostimulation with the presence of nitric acid and endophytic acid, nutrient availability with alkaline phosphatase and phytase, enzymes that talked a little bit ago, organic acids, some also enzymes like the endoglucanase, and metabolites related with abiotic stress resistance, like antioxidative enzymes like catalase and so on, osmolytes like proline and glycine betaine, and also the exopolysaccharides. During this study, we also tried to detect some genes that were related to the endophytism of this strain. So, and we detected and noted some chemotaxis genes, some motility genes, also genes that were related to the formation of biofilms that allow the bacteria to become endophytic. And we did this trial where we, in a hydroponic solution, we put some solution of these bacteria, and we wait several days to allow them to make a biofilm on the roots and to migrate to the upper part. And then several days later, we took the leaves, we cleaned the leaves with a bleach solution, and we put it on a petri dish, and some days later, we saw that the bacillus was leaving the leaves to the media, so proving the endophytism. So the second part of the study was to try to see if the bacillus improve the water tolerance on maize using the phenotyping machine that we have on our facility. It's a trait finder. So it's basically a portable machine with 2 scans with multispectral and 3D cams that allow us to monitor more than 20 morphological and physiological parameters. So as you can see, we can have also this nice data, like a 3D replica of the plants that we measure. It's a non-destructive measure, machine or method, and it's very quickly. In 90 seconds, we have all these readings. So showing you some results, like the leaf area, it's interesting to see that. So on this trial, we use potted plants of maize, and we have 2 irrigation regimes. Full irrigation, that means field capacity, and another one with a reduction of 50% the irrigation, so provoking a drought stress on those plants. And then we also have plants inoculated with the BS36 and plants that serve as a control. So on this graph, we can see that the full irrigated plants inoculated with BS36 were the ones with higher value of leaf area. But it was also very interesting to see that even with a reduction of 50% of the water, the plants that were inoculated with the BS36 presented the second highest level of leaf area. Regarding plant height, we can observe that water is crucial for the plant height, determines the plant height. Nevertheless, once again, we see that the plants in stress, with a 50% reduction of water, showed a much higher height than the control. Regarding the NTVI, we can see that the lack of water affected the physiological performance of the plants. Once again, nevertheless, the plants inoculated with the BS36 had a better performance than the control plants. Regarding the green leaf index, was the same. So at the beginning, there was already difference between the 2 irrigation treatments. But more or less, after 1 month of the study, we could saw that the plants inoculated with the BX36 showed difference to the plants of control. And in the end of the essay, all treatments have significant differences. Once again, in here we didn't use the trait finder, we used a mini pump so to measure the fluorescence. And even here, so we saw what we expected. So the fully irrigated plants had optimal values, and the stress plants have lower values. On the plants with the 50% reduction on water, we saw a significant difference on the fluorescence levels, on this FV-FM ratio, but was also surprising to see that even on the fully irrigated plants, there was a difference. So the final measurements of these trials were done, and the most important probably and the most holistic of all is the biomass, the total biomass. And once again, what we saw was that in the full irrigated plants, there was a significant difference on the biomass of the plants inoculated with the BS36. So that is relating to this plant growth promoting action that the bacteria has. And also on the stress plants with less water, we see also a big difference. And we see an increase of that biomass. So proving that some alleviation was made on those plants. Also, something interesting is that the results we got on the root-shoot ratio comply with the hypothesis that when plants are in a drought situation, they invest more on the root biomass. So as we can see on this graph, on the full irrigated plants, we see no difference, but on the stressed plants, we see that we had an increase of this ratio. That means that the plant invested more on roots, so this bacteria stimulates the natural tendency of the plants. Regarding the water leaf content was also the same. So on the 50% reduction of irrigation, we saw a difference. That probably is related to the fact that the stressed plants added a more root biomass that allows them to absorb more water. For finish, we made some leaf analysis to see the macronutrients in the plants. And once again, like expected, the full irrigated plants have more concentration of macronutrients comparing to the stressed ones. And well, on these data, for me, the most surprising is the nitrogen values that are surprisingly high there. But it's related to this plant promotion effect that we were expecting having with this strain. So to conclude, we can say that the omic technologies showed us the presence of phytohormones and enzymes related with nutrient availability that suggest the plant growth promoting effect, and also phytohormones and osmoprotectants and antioxidant enzymes that suggest the biotic stress alleviation. On the maize assay, we saw that the plants inoculated with the Bacillus thuringiensis BS36 under full irrigation, they presented a significant higher biomass than the control plants. So the hypothesis of the plant growth promoting was confirmed. Also, the BS36 was also able to promote maize plant growth under deficit irrigation. That means that the hypothesis of the drought stress alleviation was also confirmed. So I want to finish thanking my team that made this trial, and I think waiting for your questions if there is any. Thank you very much.